types of plasmids. Okay, among the types of plasmid, the most important component is plasmids coding for phenotypic traits. Okay, so plasmids are nothing but they are extra chromosomal self-replicating DNA molecules that will give special function to the bacteria. So this we have discussed earlier also. Okay, so plasmid coding for phenotypic traits means what? What is meant by phenotype? Phenotype is nothing but the physical appearance. So it should give some physical. So it should be observable character. Phenotypic trait is nothing but the observable character. There are plasmids are classified into various types based on their phenotypic traits. Okay. The plasmids which will code for phenotypic trait include, first one is F plasmids. F plasmids, which are also known as the fertility plasmids or conjugative plasmids. Okay, these F plasmids will be involved in the process of conjugation. Okay, this you have studied in the previous semesters that these plasmids, so here the phenotypic trait is conjugation. That means they can transfer themselves. From where they will transfer? They will transfer themselves from F cell to F minus cell. F plasmid can able to transfer themselves through the process of conjugation from F plus cell to F minus cell. Okay. Previously, the F plus cell is having in addition to the genomic DNA, F plasmid is present in the F plus cell, whereas the F minus cell have only the genomic DNA. It has only the genomic DNA. So F minus cell does not have the F plasmid, whereas F plus cell is having the F plasmid in addition to the genomic DNA. Okay. So what happens is during conjugation, F plus cell transfer its F plasmid from F plus cell to F minus cell, so that at the end of the conjugation, both of the cell will become F plus cell. This is one of the phenotypic trait. Okay, first category of phenotypic plasmid coding for phenotypic trait is F plasmids, and these F plasmids are involved in the conjugation process. Thereby they can transfer, the F plasmid can able to transfer themselves from F plus cell to F minus cell, so that at the end of the conjugation both the cells are F plus cell. Okay. Second important plasmid coding for phenotypic trait is Col plasmids, which is also known as the Col stands for the colicin. It's a toxic compound. Okay, the phenotypic trait over here is production of the colicin toxin. Okay, third category of plasmid coding for phenotypic trait is R plasmid. R stands for the resistance plasmid. What type of resistance they need? Yes, the bacteria need to be resisted from the antibiotic. Okay, the phenotypic trait here is they will give resistance against the antibiotics. Okay, these R plasmid will be carrying the genes such as ampicillin resistant gene or tetracycline resistant gene. So R plasmid will be having the genes responsible for resistance against the antibiotics. Okay, fourth category of plasmids coding for phenotypic trait is tall plasmid. So they will be exhibiting characteristics that is involved in the degradation of xenobiotic compounds. So tall plasmids are responsible to degrade the taurine. Okay, they are involved in the degradation of xenobiotic components. Xenobiotic are nothing but the components which are new to the biological system. Fifth category of plasmid coding for phenotypic trait is virulence plasmids. And what these virulence plasmids will do, they will make or turn the bacteria into a pathogen. Okay, previously these bacteria does not have any pathogenic capacity, but because of the presence of virulence plasmid, a bacteria will become a pathogenic bacteria. So here this particular virulence plasmid will give pathogenicity to the bacteria by producing toxins or other virulence factor. So this is regarding the plasmid coding for phenotypic